Hi everyone. For chapter 12 is this chapter is our first chapter for the nervous system. The rest of the semester now focuses on different parts of the nervous system because it is such a complex system. It is actually divided into the rest of the 6 chapters we have left for the first half of the textbook. Next semester we will go over the other half of the textbook. And as a first look into the nervous system, we will begin by talking about the nervous tissue in this chapter, both structure of a neuron and physiology of how a neuron is able to conduct an electrical signal. It's kind of like a continuation of the story from our neuromuscular junction in the previous chapter for muscle tissue. All right, so let's get started. Before we dive into the structure of a neuron, let's go through how the nervous system is organized and divided into different divisions. Organ-wise, the nervous system is very simple. It is only made up of the brain, the spinal cord, receptors, and nerves. Their main goal is also very simple. Simply put, they're just for receiving information from both external and internal stimuli, process that information as sensory information, and then send signal to initiate any necessary response in form of a command. So in a nutshell, it's pretty simple. But if we go down to the nitty-gritty details of biochemically what's going on, in order for that stimulus to get through to the central nervous system, or from the central nervous system back out in form of a command to your motor neuron, for example, is what gets difficult for chapter 12. Okay. So there are 3 main divisions in the nervous system, okay? The central nervous system, abbreviated as CNS, which includes just the brain and the spinal cord and all the nervous tissue, blood vessels, and connective tissue that are related to the brain and spinal cord. But the CNS mainly function to integrate, process, and coordinate any sensory data and sending out motor commands. Okay, so this is the main brain of our nervous system, sort of the center. That's why it's called the central nervous system. And physically speaking, it is literally located as a center of our body, just like that. So that's why it's called the central nervous system. That is for coordinating everything that comes in, and then have to coordinate anything that needs to go out in the sense of as a response. Okay. And hence, that's why there are 2 separate groups of information related to the central nervous system. Okay, so what we mean by sensory information is what we call sensory data, which is conveying information about conditions inside and outside of the body. For example, we're sensing something cold, we touch something hot, we touch something that's painful. Those are all sensory data that's coming in towards the central nervous system. Now, how your body reacts to those sensory information is what we call motor commands, which is we're controlling and adjusting those peripheral organs as a response. For example, if you touch something hot on the table, your skeletal muscles will act in effect, okay, or act in response to take your hand away from the source of heat so you don't end up burning your hand severely. So that's what we mean by motor commands, is that control or adjust in your peripheral organs as the effector tissues. If you remember from our homeostasis mechanism, we talked about receptor, control center, and effector. So these are the information that's coming in to the central nervous system and that's going out of the central nervous system. Okay, so we will talk about the spinal cord, okay? Because the thing is, we will talk, the central nervous system not only includes the brain, but it also includes the spinal cord. And in chapter 13, we'll actually go over some of the reflexes that happens automatically without us being necessarily being consciously aware of that reaction. And sometimes we have that reaction very, very quickly is because those sensory information doesn't necessarily have to go all the way up to our brain, but actually just through our spinal cord and back out. So we'll talk about some of those reflexes in chapter 13, and we'll also talk about the spinal cord in detail when we get to chapter 13. But we'll talk about the brain in chapter 14 after this chapter first, because it's kind of going from the top, the most superior portion of the body, and then radiates down towards the bottom of the body. That's the order we're going in. Okay. All right, so the second main division of the nervous system is the peripheral nervous system, or abbreviated as PNS. This includes all the nervous tissue outside of your central nervous system and what's called enteric nervous system. I'll talk about that later, but even then it's something we gloss over. We don't really dive deep into it. So there's technically three main divisions of the nervous system. Okay, central nervous system, peripheral nervous system, and enteric nervous system. So everything that is not included in the central or the enteric nervous system is part of the peripheral nervous system. So this is why you're seeing a lot of red here in the body, because it includes everything else that's radiating out from the spinal cord toward your peripheral limbs, for example, whether upper limb or lower limb. It is mainly for delivering sensory information from your peripheral limbs, for example, your fingers, your toes, and sends those sensory information towards the central nervous system, and then carry the motor commands from the brain, central nervous system, back out into the peripheral tissue. And it is through the peripheral nervous system that you can control other organs and tissues, such as muscle fiber that we talked about in the previous chapter. Okay, and those peripheral nerves goes in form of nerve fibers. Okay, and when we have bundles of those nerve fibers, this is what we call nerves. And then, so in the next two chapters, for chapter 14, we'll talk about the 12 cranial nerves that radiates out from your brain to control different parts of the body, and spinal nerves as well in the spinal cord. Now, now we already talked about the receptor, effector, and control center, and hence the receptor part, and that's doing the sensing, is the peripheral nervous system. Okay, sending that sensory information towards the central nervous system, and then when your central nervous system sends out the command, that is your effector. Effector tissues that doing the job, and then we are able to send that information from the central nervous system toward those effectors, is because we have the peripheral nervous system. Now, there are two divisions of the peripheral nervous system, two subdivisions, let's call it. Okay, so one is called the afferent division. The afferent division of the peripheral nervous system is what brings in sensory information toward the central nervous system from receptors. And we'll talk about some of these receptors when we get to chapter 15, but these are usually sensory structures that detect changes in the environment or respond to a specific stimuli. For example, we talked about Pacinian's corpuscle, we talked about the Meissner's corpuscle. Those are all sensory receptors that is for sending information towards the afferent division of your peripheral nervous system to send that sense of temperature, sense of pain, sense of pressure towards your brain. Okay, and I usually like to remember afferent, which starts with an A for arrival, A for arrival of a signal. Now, afferent and efferent is another good set of good terminologies to remember, because we will talk about other things that arrives at a node or a gland, or exit a gland, whatever it is. Things that arrive at those places, we tend to label it as afferent. Okay, so afferent always means something that are coming into the gland or whatever structure it is, so it arrives at it. So that's how I remember it. Afferent, A for arrival. And hence, the other subdivision of the peripheral nervous system is the efferent division. And efferent division is the one that's carrying motor commands from the central nervous system towards your effector, which is your muscles, glands, adipose tissue, whatever it is that responds due to that sensory information that's coming through. For example, if you're hot, your body know there's that body temperature that's getting hot, then the central nervous system will send out that command to your sweat glands, for example, via the efferent division, and tells your sweat gland to start sweating, and which is the water that's released on top of your skin surface that's able to cool you down. Okay, so how I remember efferent easily is efferent starts with an E for exit, something coming out. Okay, so that means usually it's a signal coming out or fluid coming out of a gland or structure. Those will be named with efferent. Okay, so kind of like when we talk about the kidney next semester with the urinary system, there's afferent arterial and efferent arterial, which has to do with arteries that goes in towards the kidney at the glomerulus and artery that come out of the kidney away from the glomerulus back into circulation. So that's what I mean by, we'll hear afferent, efferent multiple times with different organ systems of the body. Okay. All right. So now the PNS, or peripheral nervous system, is further divided into two different divisions, two different parts of it. I would say one is called the somatic nervous system, or known as the SNS, and the autonomic nervous system, which is ANS. Okay, we're going to expand on talking about these two when we get to chapter 15 and chapter 16. They're kind of both long in the textbook, but I summarize it into what's relevant to us or what's more interesting from those two chapters as a last lecture for the semester, because usually by the last lecture, everyone's very tired. So I save chapter 15 and chapter 16 as a last chapter wrap-up, lecture wrap-up for the semester as a transition to the next semester, because autonomic nervous system is actually something that we will talk about and use in application immensely in the second semester of A&P. Since a lot of the system we talk about is controlled by the autonomic nervous system, the ANS. Okay, we usually talk about things that what control, what turns something on when the autonomic nervous system comes into play. Okay, all right. I'll get to that in a bit. All right, so the somatic nervous system, SNS, is what controls the skeletal muscle contractions, and they can be both voluntary or involuntary in terms of reflex, which means something that we are controlling, able to control, and something that we can't control. We'll talk about both when we get to chapter 13. And then the other, like I said, was the autonomic nervous system, or known as the ANS, which is able to automatically regulate your smooth muscle, cardiac muscle, glandular secretions, adipose tissues, etc. All of these are under subconscious level, on the subconscious level, involuntary. So a lot of the stuff that has to do with the autonomic nervous system, there are things that your body is automatically doing for you without you having to think critically what to do next. That's for example, somatic. Some of your somatic nervous system is for that. But autonomic nervous system is usually for stuff that you don't need to be consciously controlling, and your body does it for you. So, and within the autonomic nervous system, there is what's called the parasympathetic division and sympathetic division, which is something we will continuously bring up the next semester, like I was talking about. For example, what will parasympathetic nervous system do to this organ or gland when it is activated, or what will sympathetic division release to activate this organ or gland in this certain situation. Both of which, usually parasympathetic and sympathetic division, acts in opposite manner to bring your body back to homeostasis, depending whether you need to turn on something even more or turn off something. So it depends. Okay, so that's why in the next semester we keep bringing back up the parasympathetic and sympathetic, is because it's all about homeostasis of your digestive system, homeostasis of your urinary system, respiratory system, cardiovascular system, etc. So that's why I like to end the first semester with the autonomic nervous system anyway, because that's what we're going to talk about immensely in the second chapter. Now, kind of like the autonomic nervous system is kind of like, what's depicted in this diagram here, contrasting between the two divisions. Parasympathetic system tends to slow down things, whereas sympathetic nervous system tends to speed up things. Okay, one gets you more chill, relaxed, and more in digestive mode. Sympathetic is more for getting everything turned on, ready to fight or flight. You're more alert, etc. Okay, and I'll talk about these two divisions more in detail when we get to chapter 15. And lastly, we have the enteric nervous system, which is pretty much just a network of neurons and nerve networks that sort of resides in the walls of digestive tract mainly. Okay, so therefore initiating and coordinating visceral reflexes. Remember, visceral means those organs, internal organs, especially those in the GI tract. And usually why we have a separate division for this control for the GI tract is because they can work on its own without instructions from the CNS. Okay, I've mentioned multiple times that the central nervous system is already busy enough on its own. There's lots going on. It's dealing with our a lot of things already. Reflexes, our critical thinking, our memory, our emotions, blah blah blah. All that stuff we're gonna talk about. So hence, there's a separate division to deal with the movement of food down your esophagus, the movement of food in your small intestine, large intestine, movement of feces, and when to release it, blah, blah, blah. Those things are, sort of things that are not, something we don't have to constantly think and voluntarily to control those things. So that's why we have a whole separate nervous system to control that area of the body. Okay? All right, so we won't be talking about this particular nervous system too much with the ENS, even in the second semester. It basically tells what the GI tract organs, what to do without having to bother our already very busy central nervous system. All right, so now we can start talking about the nervous tissue with their main cells, which are neurons. That's how we begin each chapter, and we're kind of like, with bone tissue, we talked about those different types of bone cells. With muscle tissue, we talked about different muscle cells. So now we begin by talking about nervous tissue cells. So here are some of their functional characteristics that is unique compared to other tissues and cell types. For example, neurons are long-lived, okay, because one thing they can't do is they can't do cell division. So if a neuron dies, that's the end of its life. It can't come back. We don't have, at least technically speaking, we don't have stem cells in an adult for growth of a completely new neuron from nowhere. Now, we don't know whether research later on will change that, but hopefully maybe one day we're able to find that. But that's the mystery of the whole embryonic stem cells or umbilical cord stem cells, is that they find that these umbilical cord stem cells are able to regenerate some stuff in the body. And it's still a mystery in terms of how that works, but that is something in progress of finding out what happens with those type of deal. But technically speaking, neurons can't do any division, and hence, it has to be long-lived. Once it dies, we don't expect for it to come back to life. Okay. It has a very high metabolic rate because it constantly utilizes ATP to conduct impulses, electrical impulses, for receiving those stimulus information and for relaying those commands from the central nervous system. And their plasma membrane have something special. They're called what's called excitable, meaning that we can change their membrane potential and activate an action potential for propagation of electrical signal coming through. We'll talk about that in detail when we get to that part in the lecture. But last chapter, in the previous chapter, for chapter 10, we talked about that action potential going from the neuron towards the muscle fiber. So that's the action potential that we're going to zoom into and talk about in this chapter. Now, why neurons can't do cell division is because they lack centrioles. Remember, centrioles is a cellular organelle that is very critical for cell division to occur, because they are the guys that are producing and organizing all the microtubules so that the microtubules can attach to the chromosomes correctly, and hence they're able to pull them apart correctly as well during anaphase of mitosis. But if you don't have centrioles, there's no microtubules, there's no organizations of these microtubules, and hence you can't do mitosis or proper cell division without centrioles. And that's why I said, technically speaking, neurons cannot be replaced after injury. They can be repaired. However, we'll talk about ways it can be repaired, but not 100% guarantee. It's still up to Mother Nature kind of thing to repair a neuron, but we'll talk about that process a little bit. And then neural stem cells typically are inactive except in the nose. Okay, so this is one exception. Okay, I talked about how once a neuron dies, it's never going to come back to life. The only area where we do have regeneration of neurons are olfactory receptors. We're going to talk about cranial nerves in chapter 14, and we're going to get to also chapter 17, your special senses, including a little bit about your sense of smell. And we'll talk about how those olfactory receptors, as one of your cranial nerve, your olfactory neuron, that's the only neuron in the nervous system that can regenerate over time. And that's why we can keep our smell very active. Okay. And the other area is hippocampus. Okay, hippocampus is a very complex part of the brain that is for storing memories. I'll touch a little bit of memory in chapter 15, but not too much of it, but because it gets complicated. So, but because its job is to store memories, that's why sometimes when we make new memories, we have to make new cells, maybe. Okay, so that's how those would be areas where we see neural stem cells being there and active. But I guess one wants to wonder, is it possible that one day we can use these neural stem cells to make any neurons or grow any neurons we want that we can replace? That is an interesting question in terms of future prevalences of that area. Okay. All right. So let's see. So since neurons are important for relaying electronic impulses, we have to talk about some of the cell structural characteristics that enables them to do its function. In chapter 4, we have introduced neurons by talking about these cells having a cell body, dendrites, and an axon. Okay, so now we zoom in further to look at their detailed structures. For example, there are three main parts: the cell body, the dendrites, and the axon. And we're going to talk about those other little parts, okay, within those areas. So within the cell body, which is this part right here, neuron is still a cell, so it still has those organelles that any other cell has. It still has a nucleus and a nucleolus. It still has a cytoplasm. And for cytoplasm of this neuron, we call it the perikaryon, okay? Perikaryon is only the cytoplasm of the cell body. When it gets to the axon part, we call the cytoplasm of the axon something else. Okay, we're going to get to that in a bit. And then just like any other cell, there's cytoskeleton that makes up the structure of the cell as well for a neuron. It's just that we call them, instead of intermediate filaments or microtubules, we call them neurofilaments and neurotubules, and also neurofibrils, which are just bundles of those neurofilaments that makes up the cytoskeleton of the cell body of the neuron. And they still have all those other organelles that a cell would have. You still have mitochondria, very important, if anything, because a neuron needs to rely on lots of ATP for allowing those electric signals to go through. You have lots of ribosomes because you constantly need to make what's called neurotransmitter in the neuron that we'll talk about, and how they're moved down the axon toward the axon terminal. So there's lots of protein synthesis involved in within a neuron, and of course that means there's got to be rough ER as well, because those are for your protein factories that are responsible for making those neurotransmitter. And because it's a neuron, it's something special, so they group the rough ER and the ribosomes together, and they call it bodies, okay, for the neuron. So clusters of rough ER and ribosomes together are called bodies. And why they give them special names is because these things is what gives that gray color to the cell bodies of the neuron. So I'll get to the part in the lecture where I talk about there's gray matter versus white matter. I'm sure you've heard of it in the brain and spinal cord as well. So gray matter represents those area of the nervous system that has a lot of cell bodies, which is the main cell, okay, this main part of the neuron in that location of the nervous system. And why we call it gray matter is because literally that's what it appears as. It appears gray when we slice open someone's brain and look at the layers in there. And it's gray due to the presence of these bodies, which are your rough ER and your ribosomes. Okay, so the second main part, very important as well, that extends from the cell body are the dendrites, okay? Dendrites, remember, is for receiving signal, okay? Dendrites never send out anything, okay? Dendrites are for receiving signal. So signal comes through, goes to these dendrites, any of these little branches, receive those signal, goes through the cell body, and then down the axon towards the axon terminal. Okay, so dendrites are not for releasing anything. It is for receiving signal. Okay. So these are little extensions or processes that extend from the cell body. They're highly branched, and sometimes they can be studied with what's called dendritic spines. And for example, here is a dendrite, and you see these little circles and dots there and there. Those dots are the dendritic spines where synapses happens, meaning that another neuron may come in with its axon terminal, and then it kind of sits around there and form a synapse so that it can send that signal toward the dendritic spines, and then that activates that neuron, and then the electronic signal gets propagated towards the axon and axon terminal, and so on and so forth. Okay, so dendrites are the ones that play key roles in intercellular communication for receiving signal, okay, from another neuron or from the central nervous system directly. Okay, and thirdly, we have axon, okay? The axon is very critical for conduction of an electrical impulse or action potential. It is a very long cytoplasmic process for that propagation, okay? It includes the axoplasm. That's why I said the cytoplasm of the cell body is called the perikaryon. Okay, so this cell body and the cytoplasm in there is called the perikaryon. Whereas if we're talking about the cytoplasm within the axon, it's called axoplasm. Kind of like how we had sarcoplasm for a muscle fiber. And just like how we have sarcolemma for plasma membrane of the muscle fiber, we have specific name for plasma membrane of the axon, because the axon's plasma membrane will have different sodium ion, or any ion channels. We'll talk about sodium ion channel, potassium ion channel, and those channels can have different types, whether it is on the cell body versus the axon. So that's why we call it different things. Okay, so axon lemma is the plasma membrane that surrounds the axon, and then we have this initial segment right here that connects your cell body towards the axon, and this initial segment is called the axon hillock. Okay, axon hillock will be a critical structure when we talk about initiation of an action potential, because that's the location where signal is collected to see whether it is high enough to trigger an action potential. Okay, all right. So, with these neurons, what happens is that with that axon, these axon may actually be able to branch off in what we call collateral. So this right here as a branch off of the main axon, is still part of that neuron, but it's just branching out. Remember we talked about how motor unit, for example, and how motor unit is, one neuron, and it can connect to multiple different muscle fiber. Okay, we talked about that in the previous chapter. So those could be as a result of collaterals. Okay, so this is how a single neuron can communicate with multiple cells, because they're able to split and have different feet for communicating with different things. Okay, and axons and collaterals will always end with what's called telodendria. Telodendria are these branches. Don't confuse collateral with telodendria. Telodendria is close to the end of the axon and is almost directly next to the axon terminal, like that right here. Okay, so all of these guys here are the telodendria. This is the collateral right there, as an axon branching off. Okay, not when we're close to the axon terminal. An axon terminal, of course, marks the end of the neuron, and that's where another synapse happens, and you synapse either on another neuron or synapse on a muscle fiber in the case of a neuromuscular junction we talked about in the last chapter. All right. Now, because neurons can be a very long cell, some are even a meter in length, they have special proteins that function as modes of transport down the length of their axons. Now, the movement of materials between the cell body and axon terminal is called axonal transport. And just like how we have sort of that back and forth routes for buses and train, you have the en route to Dallas and then Dallas back to Plano, for example. So you have two different tracks for that two-way traffic, okay? Same thing for our neurons, okay? They have a two-way traffic, and they have specific transport protein that travels only in one direction. So the one transport protein that allows things to travel from the cell body to the axon is called kinesin, and this direction of protein flow is called the anterograde flow. Okay, anterograde flow towards axon terminal. Okay, so that's how I could remember anterograde for going towards the axon. And then, of course, the proteins that are involved with that is the kinesin. And going back from the axon towards the cell body, which we do have those kind of traffic too, what happens is, you have neurotransmitters that are broken down and reabsorbed into the axon terminal. Sometimes those things may need to travel back to the cell body, and hence that backward traffic is called retrograde flow, moving from the axon back to the cell body. So it's kind of a reverse route. Okay, so we're going backwards. Okay, going backwards from the axon back to the cell body, and the protein that does this retrograde flow traffic is dynein. Okay, All right. So, an interesting but unfortunate side note about this traffic, this one-way traffic, is that some viruses like rabies actually take advantage of this type of transport. They will try to bypass the central nervous system's defenses by entering your axon terminal and utilizing that retrograde flow, hop on a dynein, and then catch a ride back to the cell body. Okay, so we'll talk about how our central nervous system have their own specific immune cells for fighting against pathogen. And if the virus is able to hide and hops between neurons and catch a ride with those transport proteins like dynein, then they are able to evade those immune cells by just going on and into the cell directly through that retrograde flow. And with rabies, unfortunately, that's why, once you're infected with the virus, the first thing that is affected, because they enter our bodies through and that's sort of attack our nervous system first. That's why our nervous system is the first thing that's affected when rabies virus and infection takes place. That's why the symptoms involved with rabies virus infection usually involve muscle spasms, paralysis, and even mental confusion, because it affects your nervous system directly in the beginning. Okay, and once the viral infection is established, this is a scary part of rabies, there is actually no effective treatment even as of date, and are usually fatal when symptoms have already started going on. Okay, so, but the good news is there is a vaccine for rabies, and that's why they say if you will be working with wild animals, which is their main mode of transmission through, raccoons, wild bats, etc., you have to be sure you are vaccinated against rabies. If you have, if you have just been bitten in the jungle or something, it's always better to go to the hospital for treatment immediately, just in case that wild animal was harboring rabies virus that you didn't know, and you didn't have a rabies virus vaccination. So it's better to be safe than sorry when it's too late and there's nothing else you can do once symptoms have taken hold, which means the virus infection has spread it and taken a good colony in you. Okay. So, of course, since our human bodies are so organized, there's always structural classification for stuff. So, structural classification of neurons is something we have as well. But thankfully, though, it's not too complicated. So, for neurons, they don't have any that don't have any axons. There are neurons without axons. They kind of look like this, okay, with just the cell body and the dendrites that surround it. And these are called anaxonic neurons. An usually means without or don't have, so don't have axons. Makes sense, okay? Anaxonic neurons. So they're usually small and mostly located in the brain and some special sense organs as well. But their function is still unclear. And you will hear me talk about or mention this a few times while we're talking about the nervous system, because with the nervous system, is still a lot of things are unknown. Okay, a lot of times, it is the best hypothesis scientists can come up with when we talk about the nervous system. The nervous system is one of the most complicated human systems to study. Think about just finding test subject to examine is difficult enough. A lot of things scientists want to study need to happen in a live person. A dead brain, other than structural stuff, is no good. We want to know how it functions. How do we form memories? How do this and that happens? It has to happen in a live subject, but yet we just can't open anybody's brain and look at what's going on inside. So That's why, research with the nervous system, it's difficult in that sense. And you think neurons, won't grow and repair, okay? You think that it should happen this way. We know it doesn't have centriole, so it shouldn't do anything when it's damaged, but sometimes it will, okay? You think this particular part of the brain is in charge of its function, this one function, okay? That part is just for that one function. But when you look at the activity of the brain as a whole, sometimes it turns out that every part of the brain participate in that one function. Just because there's that one part with that critical information for that one function doesn't mean that you fix that one part and it fixes that whole function. Sometimes that function is subdivided into multiple parts of the brain where they contribute evenly or in some way to that one function is trying to do. For example, like reading comprehension is something we'll talk about in a particular part of the brain that allows comprehension. But then when they look at the brain activity as a whole, when they read to someone, okay, which means as reading comprehension, understanding words, they find that the entire brain has brain activity, not just that one area that we know is important for speech and word comprehension. So that's why with the nervous system, there's still a lot of unknowns, and it's not a very straightforward black and white system. There's a lot of gray areas where we think it should happen this way, but somehow it turns out it's not. So it is a very fascinating organ system. I love talking about it, but at the same time, it can also be very complicated and unpredictable, and hence, this is something you please keep in mind of while we're going through all these different nervous system chapters. And it can change with time, okay, when we learn more from our biological research, okay? Okay, so there are three other types of these structural classifications. So there's anosonic neurons, and then there's bipolar, unipolar, and multipolar. So bipolar neurons are those with only one dendrite and one axon. They are usually rare and only present in special sense organs for sight, smell, and hearing. And we'll talk about these types of neurons when we get to chapter 17 on special senses. We'll talk about the layers of the retina inside the eye, which includes different types of neurons for their different purposes. Unipolar neurons are the third type of structural classification. They are those with fused dendrites and axon, with the cell body kind of squished off to the side like this, okay? Kind of go off to the side, and yet you have that axon again here. So that's why the dendrites is sort of directly connected to the axon there, and then the axon and the axon terminal. Most of the sensory neurons in the peripheral nervous system are unipolar neurons like this. For example, receptors located within the skin, joints, muscles, and internal organs, those have a lot of these unipolar neurons, okay? And these are the type of neurons where the axons can actually be a meter or even more long, okay? And they can go from tips of your toes all the way to the spinal cord. So that's why a neuron can be a very, very long cell, which makes them the longest cell in the body. And lastly, you have, last type of structural classification of neurons, you have the multipolar neuron, which are actually the most abundant type of neuron in the central nervous system. This is where there are two or more dendrites close to the cell body. So here's the cell body, and then you have the dendrites right around it, and then you have one axon, one single axon coming out from the cell body. Now these what these multineurons are usually residing is in the central nervous system, like I was talking about, and they are, for example, those motor neurons we talked about in the previous chapter is an example of those multipolar neurons, okay? And again, just like unipolar neurons. They can be very long, all the way up to a meter long. Okay, they have the longest axon of anything, and therefore carrying that motor commands from the spinal cord towards small muscles that help move your toes, for example, that we talked about in the muscle lab. Okay. So that's why they are more abundant in the central nervous system, because they're there to carry commands from your spinal cord to your effector tissues, whereas unipolar, therefore carrying those sensory information. You stepped on a nail, that pain come radiating through, getting to your brain, is because that's a peripheral nerve that needs to send that information towards your brain. Okay. And the functional classification of neurons are also very simple. It's just sensory, motor, and interneurons. Sensory neurons are those that form the afferent division of the peripheral nervous system. Now remember, afferent means arrival of signal towards your brain, which means these are sensory information coming in from your receptors, okay? And we already mentioned that they tend to be unipolar neurons with those afferent fibers that connects between your sensory receptors, for example, your Pacinian's corpuscle, Meissner's corpuscle, okay? And we'll talk about more of those receptors in the skin in chapter 15. But those will send information towards your central nervous system and deliver that sensation. The fact that you feel pain, you feel pressure, you feel vibration is because your sensory neurons are working properly. The second functional classification is called motor neurons, which form the efferent division of the peripheral nervous system. Remember, efferent is for exits of signal, which means these are signal coming out of your central nervous system to carry commands or instruction from your brain or spinal cord toward your peripheral effector tissues, such as organs, organ system, glands, etc. And then you have interneurons, okay? Now, sometimes a sensory neuron or motor neuron doesn't connect to the central nervous system directly, but they have to synapse on multiple what's called interneurons. Kind of like the story about the CEO of the company needing to relay his message to you through multiple chains of commands through managers and supervisors before they get to you. Same thing here. The brain needs to relay the important command in form of an electrical signal through multiple interneurons before arriving at the motor neuron to move a particular muscle as the effector that will carry out the command. So interneurons are those that are just passing the baton along until it reaches that final destination. But they are still very critical in passing that complicated information. Some of these interneurons are usually located within the brain and spinal cord, and they're there to distribute sensory information and coordinate motor activity in both ways. So they can be connecting to a sensory neuron, or they can connect to a motor neuron. And that's why they're called interneuron. Remember, inter means in between. So they sit in between sensory and motor neuron. Now, the more complex a response requires, the more interneurons you will need. And interneurons always play a part in all higher function that has to do with memory, planning, and learning, because a lot of these functions involve multiple parts of the brain, and hence sometimes you need those interneurons for relaying information simultaneously to different parts of the nervous system in order to coordinate that one action you want to do. All right. So in the tissues chapter, we talked about also identify those little dots that surrounds neurons. Remember the histology of the nervous tissue. You see these neuron cells. It looks like a star or alien I talked about. And then there's all these dots around it. And we call those dots neuroglia cells. And we briefly mentioned that they are acting as supporting cells for neurons. That's what neuroglia cells do. So now we're going to talk about the different types of neuroglia there are. whether, there's actually different neuroglia for the central nervous system and different neuroglia for the peripheral nervous system. And we'll talk about what they are responsible for and function to do. There are different actually, in general, neuroglia are those that are there to separate and protect neurons. So generally speaking, they do have that unified function, which is to, like we talked about, support neurons, give them a framework. Some actually act as phagocytes. So this is what I talked about in terms of having immune cells within the central nervous system. I'll talk about that. And then they also help regulate interstitial fluid. And then a very important interstitial fluid within the nervous system is your cerebrospinal fluid. And I'll talk about cells that are responsible for producing that and circulating that around your nervous system. So there are a total of 4 different types of neuroglia in the central nervous system. First up, we have the astrocytes, which kind of looks like this. Look like a little star thing. Okay, looks like star. Kind of look like anosonic neuron. Of course, it's smaller than that. They have a whole list of functions they perform, even though they're small, but they're not insignificant. They do a whole bunch of functions for us within the central nervous system. One important function is that they are the cells that wrap around blood vessels to form what we call the blood-brain barrier. Blood-brain barrier is what filters, your blood, so that, anything that lives or for example, pathogen or bacteria or things, proteins, whatever it is that's not supposed to be in the brain will not get through your blood vessel towards your nervous system. Okay, because we'll talk about capillaries, and one thing about capillaries is that there are types of capillaries that allow for diffusion of things. It is meant to diffuse nutrients, oxygen, all that good stuff. But within the brain, when the blood gets passed towards the brain, you want to make sure whatever blood that gets to the brain is clean. And there's an extra filtering mechanism that is achieved by having these astrocytes. And what these astrocytes do is that they kind of wrap around the blood vessel. I'll talk about that in chapter 14, but they wrap around the blood vessels with their bodies and processes and then acts as that extra filter so that big stuff can't come through into the blood of the brain, okay? All right, and astrocytes are also important for creating that 3D framework for neurons of the central nervous system, and they do some structural repairs but not to regenerate or repair a neuron's function per se, but just to stabilize tissue and prevent further injury. Okay, so not actual repair, but they do structural repair that stabilizes the central nervous system and prevent any more damage than it's already done. And I'll talk about that when we get to the repairing mechanism of the nervous system. Astrocytes are also involved with direct brain development in embryos, and they can also adjust composition of that interstitial fluid, such as cerebrospinal fluid. Okay, and the second group of neuroglia in the central nervous system are the ependymal cells. And a great clue as to what their main functions are is these little lines on these cells. If you look at these are how ependymal cells looks like, and you see these little lines. Guess what they are? They are cilia. Remember we talked about cilia. And what are cilia important for? They're important for movement of fluids around the cells. So these guys are the cells responsible for producing and moving the fluid within the nervous system, called the cerebrospinal fluid, or abbreviated for short, known as CSF. Okay, they form a layer of either simple cuboidal or simple columnar epithelium in around chambers of the brain that's called ventricles. And we'll talk about these ventricles, the different number of ventricles we have in the brain when we get to chapter 14, and you actually have to identify where they are within brain models in lab. Now, and also CSF is not only just within the ventricles of the brain, but they also travel through your spinal cord as well. And they travel through the spinal cord via the central canal of the spinal cord. Again, that's something we'll go back into detail about the structure of the spinal cord in chapter 13. Okay, so those are the locations where cerebrospinal fluid flows in circulation around the brain and the spinal cord. So that's what lines these area are these ependymal cells there for moving that central cerebrospinal fluid in one direction. There's actually a whole route of circulation we'll go through, how it goes around the brain, goes down to the spinal cord, come back up, replenish, and then go through the ventricles again, and from there it goes back out into the brain. So there's that very unique flow of circulation of cerebrospinal fluid achieved by having these little cilia for moving those fluid along for these ependymal cells. Okay, so the third type of neuroglia in the central nervous system are called the oligodendrocytes. So these are the guys here or here, those yellowish looking cells. Okay, so here they kind of form these wrappings around the neuron's axon. Now, don't mistake these for Schwann cells. Schwann cells are only present, from what we learned in lab, is only present in the peripheral nervous system. We're still talking about neuroglia of the central nervous system. And central nervous system version of Schwann cells are oligodendrocytes. Okay, and the flat areas that they form is still made up of the same stuff. Okay, so these that wrap around the axon of the neurons are called myelin. And myelin are mostly made up of lipids and other proteins, but they're mostly those same phospholipids again. And that's why they usually appear more whitish in color. Okay, but what they serve to do is they serve as electrical insulation. Remember how I talked about it's kind of like that plastic wrap around the wire. If you look at electronic wires, because that's really what your nervous system is doing, is sending those electrical signals through these electrical wires. And the neuron is like the electrical wire, and myelin is like that plastic wrapping around the electrical wire for protecting it. And also, we'll talk about this at the end of this chapter, they're important for increasing the speed of an action potential. Okay, those neurons without myelin are actually propagating signal at a much slower speed compared to those that are myelinated. And the entire wrapping length is called the myelin sheath. We're talking about all these rows of those myelin layers concentrically around those axons. Okay, and one oligodendrocyte may wrap around more than one neuron. So this is another unique thing in the central nervous system because things are in compact form. You see how this is a diagram that illustrates how it looks like in the central nervous system. And you can see how busy it looks. There's stuff everywhere, okay? And because those neurons are all, busily next to each other, that's why an oligodendrocyte can wrap around this neuron and wrap around this neuron. This is another oligodendrocyte that wraps around the two neurons as well. And here's that astrocyte we were talking about that attaches to this neuron and attaches to something else here, form that blood-brain barrier I was talking about. Here's that blood vessel coming into the central nervous system, and you see all these white feet, okay? These white little feet are those astrocytes forming an extra filter, extra layer around the blood vessel to make sure anything harmful within the blood will not travel into the central nervous system. Okay? All right, so those that are wrapped by myelin sheath, okay, we call those the internodes. So these are your internodes right here. This is an internode, this is an internode, this is an internode, okay? There are sections of where myelin sheath is wrapping around it, whereas nodes are the empty area. So this is a node, for example, the blue area that you're seeing. Those are those tiny small gaps between the myelins, or where collateral branches are able to form. So for example, an axon collateral can actually branch out this way and connect to another neuron, actually connect to another neuron or whatever it is needs to connect. Okay. Now, we talk about gray matter, and gray matter comes from Nissl bodies, which are from the rough ER and the ribosomes. Now, white matter, why we have white matter within the spinal cord and within the brain is because these are usually regions dominated by myelinated axons, because myelin is technically mostly made up of lipids. And lipids tends to appear kind of yellowish, whitish, to the eye, kind of like how we looked at adipocyte, adipose tissues and adipocytes under the microscope. So that's why they appear white as well when we look at the cross-section of the brain. So gray matter are those that contain the cell bodies, because your Nissl bodies, which is your rough ER and ribosomes, resides in the cell body of the neuron, not the axon, okay, or the axon terminal. So that's why wherever there's cell body, this area, for example, that's why they have these two different background color. That's the gray matter of the brain, and this area here in lighter tan is the white matter. And what determines whether it's gray matter or white matter is the existence of these cell bodies of the neurons that gives the appearance of gray due to existence of the Nissl bodies. And then white matters are due to the numerous amount of myelin sheath that's around these axons. Okay, and then let me erase that so it doesn't cover up other stuff. Okay, and hence microglia is the last type of neuroglia within the central nervous system, and they make up the smallest population of the neuroglia, but doesn't mean they're insignificant. They are our defense cell for the central nervous system. They're phagocytic, which means they can engulf debris, waste, pathogens. So this right here is a good example of a microglia cells, and they can roam around and look for things and engulf things if they happen to see a pathogen within the central nervous system. All right, so for the peripheral nervous system, there are just two types of neuroglia. One is called the satellite cells, which looks like this. And they surround the neuron's cell bodies in locations called ganglia. Ganglia is something we'll talk more about in chapter 13, but these are sites where cell bodies of neurons in the peripheral nervous system tend to cluster together. It's kind of like, when we talked about network of nerves, it's called plexus of nerves. So ganglia are areas where cell bodies are clustered together due to synapses that they're forming with each other. And these satellite cells help to regulate interstitial fluid around the neurons as well, kind of like what the astrocyte is doing in the central nervous system. And then the other analogy of oligodendrocyte in the central nervous system that exists in the peripheral nervous system is Schwann cells, okay? So just like oligodendrocyte, Schwann cells form these thick layers of myelin sheath around the axon itself, okay? So they form around peripheral axons because these are neuroglia of the peripheral nervous system, okay? So they kind of wrap around like little Twinkies like I was talking about in lab review, exercise review, okay? So there are little Twinkies that wraps around your axons, leaving these areas as internodes, and then those empty areas that are exposed areas of the axons called nodes. So where a Schwann cell covers the axon, the outer surface here is also called the neural lemma, other than, we talked about this being called the axon lemma because it's part of the axon. And then wherever myelin is covering up, we call that the neural lemma, okay? Axon lemma is only that immediate plasma membrane that covers the axon, for example, in nodes. and how Schwann cell wraps around the axon. Is, this is the nucleus of the Schwann cell. It forms this indentation within its cytoplasm to wrap around the axon that way, and then it starts progressing and rolling outwards in concentric circles to form this whole layer of sheath, myelin sheath that protects and insulates the axon of the neuron and really making it faster for electrical signals to travel through. All right, so just like how we've talked about what happens when muscle and bone tissues are damaged, we discuss here what neurons can do if they are injured. Okay, I talked about how we can't regenerate neurons other than those in your olfactory organs and also that in your hippocampus. But if your neurons are damaged, we can, technically speaking, your nervous system has a way to, repair it. As I've mentioned in the beginning of the chapter, neurons can't do self-division. So when a neuron is damaged, there are really very limited things that can be done. But some form of repair is possible, and usually more possible in the peripheral nervous system rather than the central nervous system. And I'll explain why that is in a bit. But in general, how well a neuron can recover, if at all, like I said, it's still kind of up to Mother Nature kind of deal. It all depends on the severity of the injury. For example, if it's a crushing injury, meaning that maybe a person was trapped in a building when it collapsed, there's an earthquake or something, and a huge stone landed on their leg and it's sitting on their leg. And, the longer that pressure is applied to the neuron, the closer it is that it will resemble a cut in the axon. So, the severity of the, neuron injury can be associated with how long it takes for that person to get help or relieve the pressure from their leg or arm or whatever it is that's trapped under pressure. Okay, so the type of repair that can happen in neurons in the peripheral nervous system usually is called Wallerian degeneration. Make sure you remember this word. It's kind of weird. Whoever named it, they call it degeneration, but it's actually regeneration of an axon if you think about the steps that it's doing, but that's what it's called, Wallerian degeneration. And here are the steps involved in this type of repair. For example, let's say a neuron was cut in half like this, like this here in the middle of the axon. So what happens first is that the axon distal to the injury site, we call this part, this is the cell body. So towards this way is proximal and towards that way is distal. So that distal stump, which is the broken part end of the axon, will then actually start to degenerate. And then the proximal stump will stay intact. Now, what stays intact within the distal stump are the Schwann cells. So this is a Schwann cells right here that's remaining intact, and a Schwann cell right there that's remaining intact. And then the macrophage is going to come in here and starts chewing up all the debris that's in the area, sort of phagocytose any waste material that is no longer necessary. And the Schwann cells that have remained intact in the beginning in step 1 here will start to proliferate. They will divide into multiple number of Schwann cells and form a cellular cord that follows the path of the original axon. So here's those Schwann cells forming. They're layering on top of each other, sort of like forming that, pathway for the axon to grow into. What we don't want happening is for the axons to start, twisting this way or twisting that way and connect to places that we don't want it to connect to. We want it to be in as original pathway as it should. So that's why the Schwann cells are acting like pathway or guidelines of light that leads the axon in the correct path and route and lead it hopefully back to the whatever particular muscle fiber is connecting to or the next neuron that is connecting to. All right, so then lastly, your axon will then grow into that cellular cord that's formed by these Schwann cells. And then once your axon is formed, Schwann cells will do their growth again around the axon and make their myelin sheath that wraps around their axon and, regenerate that whole axon part that was broken off, okay? So technically speaking, this can happen within the peripheral nervous system on its own after neural injury. So that's why, a lot of times they say it depends if your arm is going to move again or something else is going to move again or finger is going to move again or not. It depends whether this repair can happen on its own naturally. Okay? All right, so for the central nervous system, repair is a lot more complicated due to multiple reasons. For example, we already saw how busy the central nervous system is, and here's another glimpse of it. There's ependymal cells here, there's blood vessels here, there's a whole bunch of, neurons that can be very close to each other like that. So usually if an injury was to happen within the central nervous system, a lot more axons are likely to be involved just for the fact that they're, kind of crowded next to each other. If you have a pressure that's been, a trauma that's hit on your spinal cord, for example, very rarely you're only affecting one axon. You're probably going to affect a whole bunch of axons in that area. And same thing with the brain. There's, when you have a head injury from something, maybe a big blunt trauma to the head, rarely do you have just one axon that's affected due to how many neurons there are within the brain. The other thing that astrocytes do, now astrocytes are there as forming that structural framework of the central nervous system. But if they sense that there is an injury, there's some damaged stuff going on, and there's debris everywhere, they will form scar tissues around the damaged area. And their job, in a sense, is to prevent axon growth, opposite of what Wallerian degeneration is doing. Because why? There's a reason for that. Remember, we talked about how there's multiple neurons in a certain area of the central nervous system. It can involve 5 neurons, 10 neurons, okay? And because of how close they are together and how compact this whole space is already, if you try to regrow an axon within that area, what if that axon ends up connecting to a neuron that is not supposed to? And, then you're going to start making connections, okay, electrical connections where they're not supposed to be. And the effect may actually be more detrimental than not having that axon connection at all by having astrocytes forming these scar tissues to prevent any more axon growth, okay? So sometimes it's better in the central nervous system case not to make random axon growth, just to prevent it from making connections that is not supposed to make, okay? Maybe it's better that way to lose that one neuron, lose that tiny part of your sensation, than to lose everything that's part of that function, okay, around that area and disrupt everything else because now you're making connections with all these neurons you're not supposed to be connecting. Okay, and astrocytes actually release chemicals that block the regrowth of neurons as well for that very same purpose. It is to make sure that, things that are supposed to be connected remain connected, and then we're not making new connections to places where we're not supposed to, okay? All right, so it's been an hour and 10 minutes or so we're going to stop here, take a break, and we'll come back in part 2 to talk about the more difficult part of this chapter, which is an action potential.